Well, good morning. It's a pleasure to welcome you to this one-day seminar, which is devoted to master transcriptional regulation and cancer. And I think one of the ideas in choosing the speakers was to examine the function of genes that were often found as oncogenes and later found to be absolutely critical regulators of embryonal development or tissue morphogenesis. And I think this has been extremely fruitful field of research in the past 40 years or so, and has brought some very unexpected findings, both with respect to cancer biology and also developmental biology. And this is what is going to be discussed by the 8 speakers today. I'd like to thank them for being here. They'll be covering very different facets of both cancer and developmental biology. And without any further ado, I'd like to leave the floor to my friend Bruno Amati from the European Institute of Oncology, in Milan. Bruno, the floor is yours.
Thank you. It's great being here. And indeed, as you said, I think maybe one common thing between the proteins we will discuss is that some of them have been discovered as oncogenes very early on, and it's been a long path in understanding what these gene products do. So I actually dug a very ancient slide from one. This was an original, not a PowerPoint, but an original slide about the discovery of MYC. And what you see here at the bottom is that this was discovered as a retroviral oncogene, and soon after, in the 70s, soon thereafter, people realized that those genes that were transduced by oncogenic viruses in animal models, in laboratory animals, and this was in chickens, actually, in birds, were cellular genes. Of course, this is an old story. We all know that there are cellular genes that have been taken up by the retroviruses and transduced by these oncogenic retroviruses. And the other thing that was realized very quickly, and here we are in the late 70s, is that the same genes are frequently translocated, amplified, or otherwise activated in human cancer, leading to the concept that these really are driving events in oncogenesis. So we are in the 70s, 80s, and so genes like MYC, RAS, and many others were discovered like this and sort of triggered the field of cancer genetics. And we will hear a lot more about that, of course. Now, I jumped many years, and of course, a lot of attention went on to what do these genes do? How are they regulated? What is their normal physiological function and developmental function? And to cut a very long story short, just schematically, MYC is one of these genes that is called immediate early response genes. They respond to mitogenic signals. All cells that are stimulated to divide will induce this gene. And you see here a number of these signals that can induce the MYC gene either at a transcriptional level, at the level of RNA stability, or the level of protein production or stability. And this protein then participates to the response of the cell to these mitogenic signals. The cells have to divide, they have to enter cycle, they have to activate their metabolism, they have to produce energy, they have to produce macromolecules, and these genes are part of the signaling cascade that does that. Now, this particular gene product is a transcription factor. We understood that in the early 90s, so we jumped 15, 20 years now. And since then, and the other thing we understood, of course, is that this gene is required for the cells to respond, and we're talking about normal, non-transformed cells here, and mount all the responses that they need to mount in response to these upstream activating stimuli, cell growth, cell cycle, apoptosis, energy production. I will come back to some of that. Now, of course, this is highly complex, but one thing that is worth saying here is that it's the same features that make these genes such a central hub in the signaling network of the cell is what makes it a very dangerous oncogene, because, of course, it comes as a natural consequence that if you deregulate this gene in cancer, whether by any of the accidental mutations that I told you before, or in most cancers, in fact, MYC itself is not altered, but the upstream signaling pathways that you see here are mutated. They are themselves oncogenic pathways, and you recognize them here, Notch, Wnt, RAS signaling, and so on, of course, are deregulated in most cancers. In most of these cases, MYC is also deregulated by virtue of this aberrant upstream signaling and contributes to cancer progression. So Since now we are in the early 90s, since we understood that this works as a transcription factor, it's a protein that dimerizes with another protein in order to bind DNA, and I will come back to that later in the talk, we have been asking the question, what does this transcription factor regulate in order to implement its biological activities, either in a normal context or in a tumor context? Now, of course, this we need to put back in the context, and this is a few examples of genes that have accumulated over the years. This is a small list here, and this has been evolving over the last 20, 25 years. Maybe there's a note here that is useful making. I think for all of us working on oncogenic transcription factors or transcription factors in general, the question of what does this gene regulate in order to fulfill its biological function has been one of the most elusive questions throughout the years, and for two reasons. One, the system is very complex and there's many indirect feedback mechanisms. Two, the technology has been limiting for many, many years, right? 20 years ago, we identified in a gel retardation assay one promoter that was bound by the recombinant MYC protein, did a luciferase assay and showed that this is activated. You got your cell paper. Today, you need the whole genome, and this may not be enough in most cases. So the field really has been evolving with the technology. If we consider what we were publishing and the tools we had to address those questions 20 years ago, we almost feel it was prehistory, and yet it's only a couple of decades. So I have no time to go through that in too much detail, and I will try to give you an excerpt, essentially, of where we are today. What I want to distinguish here, in order to discuss this, is the situation of a normal cell that will be activated by mitogenic stimuli. As I told you, MYC is one of those genes that is induced. This cell has to enter cycle, has to divide, has to synthesize macromolecules, and of a cancer cell that overexpresses MYC and will have further acquisition of features that are aberrant in cancer. And one of the sources of confusion in the field is when we compare normal and transformed, frequently the comparison when doing is a cell which is a normal cell in this state with a fully transformed proliferating cancer cell. So there's many things happening there, and frequently we have to put them all in the same bag because we don't really distinguish between those two steps. And so we have, and others have tried to distinguish those things carefully over the years, and I'm going to be extremely schematic here. When you activate a quiescent cell in any tissue by any means, usually you have a bunch of genes that come up, Jun, FOS, MYC, other early genes. None of these are transcription factors. The model would be that these induce a series of target genes that then contribute to all of these events that have to happen in the cell. So there's a transcriptional cascade here that responds to the signaling cascade that was triggered by membrane receptors in particular. Okay, now we don't have time to discuss this in much detail, but in 2012, two papers came out that proposed a very different view of how this transcription factor might be working, proposing that it's not really a selective transcription factor, but a global regulator of transcription that will directly lead to this global increase in RNA biosynthesis that has also been called transcriptional amplification. Again, this is a debate that is still ongoing in the field. I should say that there's a lot of disagreement on this issue yet, so I am not a neutral part on this, of course. What I will argue here, and I'll give you maybe a few examples as I go on, is that all of the data we have, all of the elements we know, are explainable by virtue of this scenario here, which is that these transcription factors regulate selective, still many genes, but selective sets of genes that then contribute to those cellular responses. And a few of the relevant references are here. Again, we're not going full detail here. One of the questions, of course, is, are we able to identify such genes that are induced by mitogens when we activate cells in a manner that depends on the activation of the transcription factor in the first place? So we did this type of experiments quite carefully. Some of it is published, some of it is unpublished. I'll show you some of the unpublished data by taking cells, put them in culture, make them quiescent, so G0 cells, quiescent cells, and activate these cells with mitogens in two conditions. One is the control cell, which has MYC and will induce it, and the other is the same cell in which we have induced deletion of the MYC gene in the quiescent cell before activation with a mitogen, with a growth factor. So that cell will not be able to induce MYC, and we'll address whether there are sets of genes that fail to be induced or are induced in a lower manner, in a weaker manner, in the absence of the transcription factor. And indeed, these genes can be identified. What you have here is a dot plot that comes from an expression profiling experiment, so RNA sequencing experiment, in which we have compared those two populations. We're looking at the response of genes to the mitogen in the cells over a few hours. In wild-type cells on one hand, so if something here is above 0, it means it's induced. Below 0, it's repressed. This is a log2 fold change scale, fold change in gene expression in a log2 scale. In the y-axis, you have the same data for the mutant. So if a gene is induced independently from MYC, it will be here on the diagonal, of course. It will have a positive value in the wild type, a positive value in the mutant. And what you see here right away is that there's a large part of the transcriptome that is normally regulated, so it's along the diagonal, but there's a quite significant group of genes that are regulated less or lose regulation, induction in this case, in the mutant compared to the wild type. And there's a bunch of genes here that are repressed in the control cells, but are less so in the mutants. So this transcription factor seems to be required for those changes in part of the transcriptome. And of course, the question is, what are those genes? Here you see a heat map which visualizes this. This is the induction, those induced genes in the wild type, in the knockout. You see induction here, which is lost, and the same thing for downregulation. The question is, of course, what are I will come back to what those genes are. What is the timing of all of these events? And what I want to show you here is that these events are quite rapid. In 2, 4, 8 hours, we see these responses. And this precedes any sign of activation of the cell at the bulk level. In particular, it precedes this global increase in RNA biosynthesis that I mentioned before, and it precedes the increase in cell size. So these transcriptional programs are put in place and contribute, most likely, to the growth response of the cell, which includes an increase in macromolecular biosynthesis, RNA, protein, and many others. And indeed, if we look at the function of the genes that are induced in a MYC-dependent manner, we find many things that are involved in protein biosynthesis, in particular ribosomes, things that are involved in transcription, things that are involved in energy metabolism, mitochondrial biogenesis, other metabolic pathways, biosynthetic pathways, and so on. So this fits with a scenario in which there's a transcriptional cascade here that sets in place the accumulation of gene products that drive those changes in the physiology of the cell. Let's consider the second part for a minute here. So if we now take a cell which is proliferating and overexpress MYC in an acute manner in that cell, what we see is that MYC levels will increase, of course, now to pathological levels. If we do chromatin association by chip sequencing experiments, genome-wide, what we see is that there is very general interaction of the transcription factor with regulatory elements in the genome. I have no time to show you detailed slides, but most of promoters and enhancers that are active in any given cell type will be cross-linked and detected in this type of assay. So this is one of the things that has led to this idea that this is a general regulator, and I will come back to that. And then what we see in most cases is that there is selective responses of genes again, which are overlapping but distinct from the ones that were induced in the first place. There's some of the papers that have addressed this are here. And in the opposite manner, if we take a tumor and shut down MYC in an acute manner, this has been done again by a number of laboratories, we see that the transcriptional response is partly reverted. There is also in here some disagreement between the conclusions that are made by all of these papers. Again, I don't want to go in detail of every one of these, but the one conservative interpretation again here is that what we see is that the acute activation or inactivation of MYC in tumor cells drives the response of a defined, albeit large, transcriptional program. Okay, we'll give you one example here. Here we're taking cells that are proliferating. These are mouse fibroblasts that express a MYC estrogen receptor chimera which is a fusion between MYC and the hormone binding domain of the estrogen receptor. So now this protein is inactive, it's expressed in the cells, but it can be induced by ligand, by estrogen in particular, or estrogen analogs, in this case, hydroxytamoxifen. And what you see here is that over time, when we induce with the activity of MYC-ER in those cells from here to here, we get a significant number of genes that go up, significant number of genes that go down. This is in the absence of any global change in transcriptional activity. So again, we have a selective response. And the lane here is newly synthesized RNA that we sequence alongside the total RNA. And in particular, we do this by a short pulse of 4-thiouridine, which gets incorporated, allows to biotinylate the newly synthesized RNA and purify it, and sequence it alongside the total RNA. And you see that most of the change that we see here at the total RNA level are reflected in changes in synthesis, and I will come back to that. The question comes when we look at where does MYC bind in the genome and how does that compare with the activity of those genes. So trying to correlate the binding of a transcription factor to a locus with the regulation of that locus. And this seems simple, but has turned out to be much more complicated than anticipated. What I show you here is the increase of MYC binding to a series of promoters. So each row here is a promoter. Each column is a data point, is a time point. And when you see red here, it means that the corresponding promoter was called as bound by the transcription factor in a cheap sequencing experiment, right? So those are all the promoters on chromosome 1 here in these mouse fibroblasts that I showed you before that are bound by MYC sometime during the experiment. And what you see here is a serum stimulation, 0, 1, 2 hours. So these MYC levels are increasing here. You see it on more and more promoters. And then we activate MYC-ER on top, as I told you. So this is the exogenous protein, binds to many more promoters, right? And then we look at what those promoters are, in fact, which is quite striking. And I think all of the data agree on this from many, many laboratories. At the end of the experiment here, we're cross-linking MYC to all of the active promoters in the genome, virtually all of them. And what this shows you, again, I will not go into detail, it's a bit confusing, but it's all red, as you can see. And what this shows, what this is shown here is the sequencing patterns for chromatin immunoprecipitation experiments, again, with markers of active transcription, of active promoters, RNA pol2 itself, and then histone methylation events or acetylation events that correspond to active promoters, H3 lysine 4 trimethylation, monomethylation, H3 lysine 27 acetylation, and this is DNA with 1 hypersensitivity. So we're producing all these data sets on the same cells in the same experiment in the same time course and comparing the state of those promoters before and after activation of the transcription factor in the same cells where we have looked at the profiles of the transcription factors itself. And what you can see here is that all of these promoters that are bound by MYC are promoters that were already active. It's already here in the cells before we activated the transcription factor. If we look at the other side of the coin, that is all of the promoters, because we have annotation of all the promoters in the genome, of course, we can track all of the promoters that are not bound by MYC in this experiment. And you see that this is white not only for MYC, but it's virtually all white here as well. So these are inactive promoters. They are not marked by polymerase II, by and large, or by the active histone marks. Most likely they would be marked by repressive histone marks if we did all of the profiles. Okay, so what I'm telling you is this transcription factor goes and binds to active promoters. I'm not showing just a note here. We see the same thing on enhancers. So this is what I'm showing you here is proximal promoters that are annotated transcription start sites. The same thing is true at distal sites that qualify as enhancers. They have all of these chromatin marks as well, and so on. The question then is, how do we go from this, which is a very widespread association with the genome, to this, rather selective transcriptional response, right? This is part of the confusion in the field between the idea that it's a global regulator or a selective regulator. One of the issues that we asked is whether it would be the level of binding, that is the gain of MYC binding at promoters, that would be associated with these responses. And to look at that in more detail, we take the same data set, and here we look at the relationship between expression changes, so these will be repressed genes, these will be activated genes, at a given time point. This is 4 hours after MYC-ER activation. And we look at the ratio of MYC binding to those promoters before and after activation of the transcription factor. So 0 would mean no change in levels, and the positive value here means that you have more, because we are activating more, a larger amount of the transcription factor from the control cells, which are proliferating with endogenous levels of MYC, to the ones in which you have superactivated the protein. And if we take the same data set and express it in a slightly different way, here we're looking at what we call the MYC share, that is the ratio, the relative gain or loss of MYC from those promoters. Remember, it goes up in those cells, so you will see more everywhere, right? But the relative amount is changing, and some promoters have a stronger gain, some promoters have a lower gain. So if you normalize this to the total amount of the transcription factor bound to the genome, some of these will have a decreasing share, some of these will have an increasing share. And increasing this MYC share actually correlates pretty well with activation. Decreasing it, that is showing a relative loss of the transcription factor, even though there is more bound everywhere, corresponds to loss of transcriptional activity. So the question is, how does this work? I will skip a couple of slides here because it's not necessary to go into full detail, but just to show you that this is a highly conserved feature in a number of data sets where the data are available. We analyze all of those data sets in the same manner. That MYC share actually is an excellent predictor in most of these cases, in all of these cases, of the relative transcriptional response. This is a different way to show those data. I will not go in detail. So when we saw this, we decided to try to explore this in more detail mechanistically. And what we decided to do is to take the same, our model system, the same fibroblasts here that I was mentioning before, and go into a more detailed kinetic analysis of the changes in pol-2 distribution and transcriptional activity through time after MYC activation. So this is the scheme of the experiment. We're going to do RNA sequencing of total RNA, of newly synthesized RNA with the trick I told you before, the 4SU sequencing. We're going to profile MYC by chip sequencing, and RNA polymerase II by chip sequencing. And we're going to do this, all of these time points here, that is very narrow time course, 0, 10, 20, 30 minutes, up to 16 hours, as shown in this slide. With these precise and quantitative data in hand, there's a number of things we can do. We can model the life cycle of RNA in terms of the transcriptional activities. In particular, we can model the kinetic rates, and this has been developed in the lab of Mattia Pellizzola, with whom we have collaborated, this is the paper they published with the method. So the kinetic rates can be modeled. What do I mean by kinetic rates? I mean synthesis rate, processing rate, and degradation rate for every pre-mRNA and mRNA in the genome. And when we do that, what we see, and we can plot it here as a heat map, is that this is the mature RNA from the RNA sequencing experiment. This is pre-mRNA, and then the three kinetic rates: RNA synthesis, processing, and degradation. And what you see here right away from this heat map, even without going into any detail, is that the one that correlates the most closely with total RNA changes is synthesis rate. This tells you simply that all of these gene expression changes occur at the level of RNA synthesis, that is transcription, and with relatively little, although there are interesting details here, but relatively little contribution, at least at the global level, from processing or degradation of the mRNA. Having seen that, we can now take the synthesis rate and compare that with the behavior of RNA polymerase II. So now we are going to align those RNA data with chromatin immunoprecipitation experiments, for the chip sequencing experiments. So we have RNA polymerase II. We can quantify it in the promoter of genes, the gene body, or the transcriptional termination region, as you see here, this schematically, right? So we quantify those data. We look at the changes in the amount of pol-2 in those regions of those genes, and we calculate 4 other parameters which can be incorporated into the model. That is what we call the flux, but essentially this is the loading of polymerase II on promoters. Post-release, as you know, polymerase II is posing most frequently after initiation and needs to be released from this step, and there's a number of regulators that release this. Elongation and release from the transcription termination side, of course. Again, the formulas are here. Again, this was done by Mattia and Stefano De Pretis, Mattia Pellizzolo and Stefano De Pretis. With this in hand, we actually can model those steps. We can define a number, still on this here, we can define a number of clusters of genes that show similar behaviors in these data sets and look at how those parameters contribute to transcriptional control. And just to cut a long story short here, this is the different clusters that I showed you before, induced genes, repressed genes. What this slide shows is that the first step of control is loading. That elongation is also regulated, but seems to be ancillary to the initial loading step. And again, there's a very hot debate in the field here about whether MYC regulates elongation or loading or both. Long story short is probably both, and they need to be integrated into in a precise manner, and this is still to be investigated. The most important thing here, I think, and there's a lot of this type of activity in the field, is that really this kind of dynamic changes, identifying the critical steps in the transcriptional cascade, cannot be done by simply looking at steady-state distribution of RNA polymerase II in two conditions and then calculating the ratio between the promoter and the gene body, which is what most papers do. And this is what I call here, what by this so-called RNA stalling or elongation index that you will see in most papers. Now, the other thing that this I can skip, essentially it's a confirmation that the MYC share correlates not only with expression, but of course with the level of Pol II loading. So if I summarize this into a schematic slide, I've shown you that when we take a proliferating cell that grows with MYC is expressed in those cells, but at relatively low endogenous levels, and we superactivate this, the transcription factor, as would happen in a cancer cell, a number of genes are induced, a number of genes are repressed, and this correlates with the relative changes in MYC binding. MYC binding increases everywhere, but if we express this as the relative share between all those promoters, we see that the increase in the levels of MYC in this MYC share correlates with activation, and the opposite is true for repression. So MYC binding intensity per se seems to be a determinant of the transcriptional response. Of course, this will not be sufficient to express the full thing. I should just say that there's a number of similar observations in a number of recent papers which are cited here. Again, without going into detail, if anybody's interested, we can discuss this later. The other thing that is important here is that to try to understand what happens in terms of repression, because I told you most of the repression might be indirect. In fact, the repressed genes are the ones that show a relative loss of the transcription factor on their promoter, which is counterintuitive if this was acting as an active repressor. Whereas in the field, clearly there are repression mechanisms that have been described and demonstrated on individual genes in the past, right? So we need to incorporate this into the picture. And we need to determine what fraction of this repressed transcriptome is repressed directly as opposed to indirectly. And one corollary of what I told you here, of this indirect repression, which is something I would like to throw out as a concept that is a bit provocative, but for which we have no evidence whatsoever, is that maybe those repressed genes are genes that are, rather than being actively repressed by MYC, by the transcription factor, do not compete efficiently for limiting amounts of polymerase II in the cell. So how limiting is RNA polymerase II to sustain all of the transcriptional responses that a cell needs to sustain, and how many genes are repressed as a consequence of not being effective competitors for RNA Pol II is something for which there is very, very little evidence in the field. And I'm not aware of any definitive data that would address this question. Okay, now the second, which I told you concerns the mechanism here, and I think I just already made the point. So this is a summary slide that summarizes what I told you so far. Let me skip it for the detail because I really want to go on with some of the forthcoming issues that I want to discuss. Among those questions that I think are open for the future, I will want to discuss two very briefly with you today. One, of course, I told you that the transcription factor binds everywhere in the genome. By chip sequencing, you see it cross-linking everywhere. I told you that those genes that respond are the genes that are bound the best. This, of course, is a sequence-specific transcription factor, so presumably sequence recognition, which corresponds with higher affinity, should play a role in this transcriptional response. This is one of the questions we decided to address in more detail. And then, of course, the other is what is the contribution of this global binding to regulatory elements, enhancers, or promoters in MYC-driven transcription. So very briefly, if we consider the structure of this transcription factor, I told you it acts as a dimer. Its dimerization partner, the only one that is known in the cell, is called MAX. They are both basic helix, subhelix, leucine zipper proteins, so they have this 80 amino acid consensus motif that is a motif required for dimerization and DNA binding. So this has been established for a number of proteins. You know many others, MyoD, for example, neurogenic, myogenic factors, tissue determination, transcription factors, and a number of others. They form specific dimers with specific subsets of these proteins, and they bind variants of this so-called E-box consensus element. The crystal structure of this dimer, just this region here, bound to DNA, has been resolved in the past, and it's actually quite a beautiful structure. What you see here is that the protein needs to dimerize via this HLH zipper domain here to present the alpha helix here, or the basic region, in the scissor conformation in order to engage it into the major groove of the double helix, right? And when you are doing this, the specific contacts that are established and non-specific contacts that are established with the DNA. Some of these contacts in the basic region are between arginines that form phosphodiester bonds, that form salt bridges with the phosphodiester bonds, so positive, negative charges in the backbone, and some contacts are sequence-specific with a consensus element. In particular, this histidine within the basic region and this glutamic here form base-specific contacts that determine binding specificity. So what we decided to do was to try to mutate either the non-specific contacts with the backbone or the specific contacts. So this mutant here has the 3 arginines mutated to alanine, and this mutant here has this histidine and this glutamic mutated to alanine. And we did a lot of work with these mutants to characterize them. Again, I will show you only a couple of slides here. We express them in cells, and we try to understand what they regulate, what they bind, and whether they're active or not. This is a cell system in which we have MYC expressed with a tetracycline regulatable promoter, so that we can switch MYC off in those cells. The endogenous gene is inactivated, and so the cells depend on the exogenous TET-MYC gene. And in those cells, we can superinfect them with a retrovirus that expresses the wild type, the specific mutant, or the generic DNA binding mutant, and ask, what do these mutants do? The first thing is that if we withdraw doxycycline, which suppresses expression of the TET-MYC gene, then what you see is that the only cell population that keeps growing is the one expressing the wild type. The mutants cannot do it, or the empty vector cannot do it. So they are biologically inactive. If you lose sequence recognition, you lose biological activity. If we look by chip sequencing in those cells, what is the binding pattern? This is after suppression of the exogenous TET-MYC molecule. So we're looking at the binding by these transcription factors here. Just focus here. This is the peak calling. This is the quantitative heat map. It's the same data set. Again, each row is a promoter. Each column is a sample. And so at the top here, you have the promoters that are bound in the most significant manner, down to the ones that we call are less bound. And what you see here is that in this particular scenario, the mutant that loses the contact with the backbone is completely inactive. We detect it nowhere on chromatin virtually. The mutant that loses sequence recognition still shows some binding, some generic binding. And I have no time to show you, but indeed, those sites do no longer enrich for the consensus site. We decided to try to make a more physiological experiment by knocking in the mutant into the cell. So we're using the same cells again, but instead of overexpressing the mutant, we knock in the mutation into both of the alleles of these cells. And what you see here is that whereas wild-type MYC is still showing binding, of course, which is the same as here, binding by the mutant here is completely lost. And we don't detect it anymore. I'm going to try to put this into what we think this means into a schematic context of what a transcription factor has to do when it needs to recognize its target sites in the genome. It needs to engage on open chromatin via protein-protein interactions or accessibility, or compartmentalization in the nucleus, of course, which is an important parameter not discussed here. It needs to engage on DNA in a non-sequence specific manner, and then it needs to scan around and eventually get stabilized on the specific consensus element. And what this order of events, if what we mutated managed to mutate with this MYC-RA or MYC-HEA mutant, this one is unable to bind, and the consequence is that it does not engage on chromatin anymore. It's stuck here at some point, but at least by ChIP-seq, we don't detect it, right? The other mutation, which engages on DNA, does it only non-specifically at this point, cannot transition to a more stable state when it finds the specific sequence, so it never finds a specific sequence. And what I'm telling you essentially is that this is not productive for biological activity. I showed you the data that it doesn't substitute for endogenous MYC in cells. I'm not showing you all the RNA-seq data for the sake of time, and this is also very recent data. We're still analyzing it. But the essential conclusion here is that it still binds to DNA and invades active chromatin when overexpressed, as I showed you. It does not enrich E-boxes, and it loses the ability to regulate those genes that are regulated in a MYC-dependent manner. Okay, so in other words, and I think the most important message here for all of us working on transcription factors is it's not because you're detecting a protein on a promoter that is actually doing a job there. In this particular mutant is still binding to all of these promoters. It's not regulating them in a normal manner. Okay, this brings me back to the beginning. I think I've got 5 minutes still, or yes. Unfortunately, I've been a bit slow, and this is a really important part of my talk. So the real questions are here. After having discussed all this, we're trying to map the transcriptome that is regulated by MYC and understand what it regulates in cell. Which of these genes are really important for tumorigenesis? Which of these genes are potential therapeutic targets in MYC overexpressing tumors? This is really the scope of all of this research. Very, very quickly, this is again one of the examples of transcriptional profiling here in a lymphoma model that is driven by MYC, comparing control with pre-tumoral with lymphoma B cells. Some are induced, some genes are repressed. We took a bunch of induced genes. We went through a functional screen using reverse genetics, shRNA screening in this case, looking for genes that would be critical for tumor maintenance. Let me not go through the detail of this slide, but the outcome of this is a group of genes here that are candidates for genes that are induced by MYC and are required for tumor maintenance, and therefore could be interesting therapeutic targets. Among those, we found components of one particular cellular complex, which is the mitochondrial ribosome. Again, this is a schematic slide, which I don't want to go into much detail, but all of the components of the mitochondrial ribosome are encoded by nuclear genes, and the majority of these genes are bound and upregulated by MYC, suggesting that MYC needs to upregulate the activity of the level of mitochondrial translation. And indeed, if we intervene with this function either genetically or pharmacologically, and we can do this because a number of antibiotics, here I showed you tigecycline, which is a tetracycline analog, have inhibitory activity against the mitochondrial ribosome. And this is due to the fact that the mitochondrial ribosome is evolutionarily and structurally related to prokaryotic ribosomes, and so these antibiotics cross-react. And in this paper that I show you here, back-to-back with another one from Steve McMahon's lab, we showed that there is synthetic lethality between MYC activation in cells and inhibition of the mitochondrial ribosome with tigecycline. We became interested in addressing whether this particular pharmacological sensitivity would be exploitable in a given tumor context. And I don't want to go to the detail here again for the sake of time. We decided to study double-hit lymphoma, which is a subset of non-Hodgkin lymphoma that is highly aggressive and has activated two oncogenes by translocation, MYC and BCL2. When MYC and BCL2 are both overexpressed, there is massive lymphomagenesis. This has been shown in mice over the years. And of course, the subset of patients that come up with these so-called double-hit lymphomas that show both translocations are the ones that respond the worst to all frontline therapies that clinicians have today. So this is a missing need in the clinic. We decided to try to go and address it. Let me skip a few slides here because they address the in vitro biology. Just show you this. This is a double-hit lymphoma cell line. If we treat it with the antibiotic alone, little happens. If we treat it with the BCL2 inhibitor alone, which is called ABT199 or venetoclax, little happens in terms of cell viability. If we treat it with both, there is a very clear synergistic activity between those two compounds. We have all kinds of control here to show that this is on target, that this is happening because BCL2 is being inhibited by the compound, and it is observed in a BCL, in a double hit, but not in cells that don't overexpress BCL2. With this, we went in vivo. Again, let me skip some of the in vitro data. In vivo, either with subcutaneous tumors with the same cell lines. What you see here is that single treatment doesn't halt tumor progression significantly, either with the antibiotic or the BCL2 inhibitor alone, but the two together do. And most of these mice actually are cured in the long term. And the final data that I want to show you here is patient-derived xenograft of a double-hit lymphoma. Again, this is the progression of untreated mice, and this is quantified by luminescence. This is single treatment with the antibiotic, single treatment with the BCL2 inhibitor, both together. We have a very clear cooperativity in terms of tumor shrinkage. Okay, so what I've shown you here, essentially, is that through all of these genomics and genetics, we came up with a few candidates, and of course, many others in the field have done this type of thing, and that some of these actually show therapeutic potential in MYC-driven tumors. And in this particular case, this is interesting because the two compounds I told you about are drugs that are already in the clinic. So we are now proposing to repurpose those two drugs against this particular subset of very aggressive lymphomas. And we're actually designing a phase 1 trial at the moment on this. Okay, this would be the end of my talk. I had a couple of things that I had at the back here as a backup to give you some idea of future perspectives. Maybe I should skip this at this point, Hugues, and just go. I want to mention maybe only one of these. So this is, we're trying to design a better model of double-hit lymphoma in the mouse, and I will skip this, but essentially this is the goal, to try to pursue this preclinical studies in a more advanced manner in an immunocompetent model of double-hit lymphoma. This is what I skipped. The other thing we're trying to do here is to screen for compounds that, like the antibiotic, would synergize with the BCL2 inhibitor to kill those double-hit lymphoma cells. And this is just one slide from a very recent pharmacological screen that I want to mention because we did it in the collaboration with Oscar Fernandez Capetillo's lab at the Karolinska Institute in Sweden. The compounds you see at the top here are compounds that show strong killing activity when combined with the BCL2 inhibitor in this pharmacological screen, where we screened in a high throughput format about 4,300 compounds. We're still deconvoluting this to actually address the synergy between the two compounds. But I think this gives you a sense of where we're trying to go, coming from all of these mapping of MYC binding transcriptional profiles and genomics to preclinical models, and to eventually identify new candidate drugs that may be translated in the same way in the clinic against this very particular subset of aggressive lymphomas. I run out of time. Let me put up the most important point here. All of the people who helped and collaborated with us all throughout the years, let me just leave them and acknowledge them here collectively. Without them, this would not have been done, of course. Thank you very much.